Welcome to the PHC Corporation of North America. This webinar video with Product Specialist Holly Hattaway addresses new tools and the evolution of 3D cell culture. Here, Holly discusses models associated with traditional cell culture methods and compares outcomes with 3D or spheroid cell cultures now possible with new PrimeSurface brand low-adhesion culture plates available from PHC Corporation.
The title of this webinar will be Adding a Dimension to Your Cell Culture. Since the early 1900s, cell culture has been utilized for studying animal models on a cellular level. And traditionally, a single sheet of cells adheres to the plastic or glass surface of culture ware and forms a single layer where some cell-to-cell adhesion and interactions occur. Although this method has broadened our understanding of many mechanical and physiological cellular mechanisms and pathways, its limitations are clear when you consider the cell's in vivo state. And simply put, cells don't grow in a single sheet. So that being said, a lot of cellular interactions that occur in vivo do not in the single layer of cell culture. Not only do cells in this single two-dimensional model not demonstrate an accurate in vivo morphology, it does not have an extracellular matrix. Now, the extracellular matrix, or ECM for short, it's an important biological component present in all cellular tissue. Not only does the ECM provide support and anchorage that helps determine the overall shape of cells and tissue, it also regulates a cell's characteristics and its behavior. This includes the cell's survival, its proliferation, polarity, and its differentiation, ability to adhere, and migration. So because of this, a central focus of cell culture has been the development of models that better mimic in vivo conditions. One of the best known models for this is three-dimensional cell culture, often called 3D culture or spheroid culture. 3D cell culture has continued to gain a lot of popularity in many labs throughout the world because it's more in vivo-like characteristics and behaviors. And as its name suggests, 3D cell spheroid models are able to grow and interact with their surroundings in all three dimensions. Because of this, 3D culture can yield more biologically relevant and consistent research results. And because of the physiologically relevant morphology, the diffusion of nutrients and gases act as it does in vivo. So more specifically, the nutrient and gas gradients form as they do in real life. This is an important factor to be considered, especially in tumor growth or drug discovery and other metabolic applications. And there are many commercially available culturing tools for 3D cell culture formation. They can be separated into two main types of categories, the scaffold-based and scaffold-free techniques. So in scaffold-based 3D culturing, the scaffold is intended to mimic the extracellular matrix. Within scaffolding technique, there are two main types of scaffolds. There are the naturally derived scaffolds, and there are the synthetic or the polymeric scaffolds. So the polymeric-based scaffolds or synthetic ones use synthetic hydrogels or other biocompatible polymers that are biologically inert. And these types of scaffolds can generate the physical support for 3D cell formation. The scaffold layout needs to match the tissue or the cell line of interest in reproducing its structure, its scale, and its function. So this requires a skilled technician and extra time to properly execute those fabrication steps for the desired extracellular matrix characteristics, depending on the cell type. The other type of scaffold-based three-dimensional culture uses matrigels or biologically derived scaffold systems. These provide the physical support necessary for cells to attach and reorganize into 3D cultures. The benefit here is that the ECM, the extracellular matrix, does not have to be manually fabricated in order to mimic the proper endogenous ECM. But the major drawback here is that they also can contribute to soluble growth factors and hormones, along with some other identified components that can affect gene and protein expression. So therefore, it can affect relevant outcomes in your research. Now, the other type of 3D cell culturing is referred to as scaffold-free. This technique allows the cells to self-assemble. They form a non-adherent cell aggregate called spheroids. And these spheroids mimic in vivo cellular tissues by secreting their own extracellular matrix. They also, due to their overall morphology, display differential gas and nutrient availability. And there are many types of scaffold-free culturing methods. Some of them are well-known ones, like the hanging drop or the agitator methods that I have pictured here. And they've been utilized for a long time, some for decades. These methods are known to encourage cell aggregation and spheroid formation. But in the case of applications like staining and imaging, or applications that require specific concentrations of a variable, these methods can be difficult or can't be performed. And it's also difficult to achieve size uniformity with these methods, which is particularly important for obtaining reproducible results. Because as most of us know, cellular functions within the spheroids is strongly linked to the overall spheroid size. Okay, so for this reason, the scaffold-free cell culturing method that I'm going to focus on here will be the low adhesion method. And this is done by way of ultra-low attachment plates. They're often referred to as ULA dishes or plates. And the ULA plates that I'm discussing today are PrimeSurface ULA plates. So these are plates that are coated with a unique ultra-hydrophilic polymer that is covalently bound to the plastic plate surface. So this enables the cells to aggregate without adhering to the culture itself, providing a stable, non-cytotoxic environment and without material degrade. They can also be seamlessly utilized for things like imaging and other applications without any added transfer steps. These ULA plates, rather than other 3D culturing methods, have shown to be very adaptive to existing laboratory protocols, which is nice. And they also can be utilized for imaging in their clear plates or for chemoluminescence screening in their white plates without any added, again, transfer steps. And so spheroids grown in low adhesion culture plates are consistent, again, in their shape and size. And as I've mentioned before, this is particularly important when obtaining reproducible results because the cellular functions within the spheroids, again, strongly correlate with their size. So here you can see two different breast cancer cell lines. These were grown in the U, M, and V-shaped bottomed wells. So you can see the image up there with the different shapes. Now, as you can see, with that V-shaped wells, the steep-shaped bottom supports the weak aggregation of these cancer cell lines, and it permits a tighter spheroid formation. Excuse me. So researchers have found that the V shape works well with cell lines that demonstrate weak aggregation, while the other shapes work well depending on the cell line and its affinity for aggregation. As well, there are in applications that require like long-term culturing, media aspiration and dispensing has to be done extremely carefully to avoid disrupting these unattached spheroids. In this case, culturing of spheroids can be made easier using a split well design 96 well plate, an ultra low attachment plate. These 96 well plates allow the media to be aspirated in one single step rather than disrupting spheroids. This just decreases the overall pipetting time by like over 80%, and it minimizes the risk of disturbing the experimental spheroid or damaging anything there. Now, when it comes to the stem cells, prime surface plates play an important role in producing uniform spheroids. So again, this is about the size. The stem cells cultured as spheroids are known to be greatly influenced by the size for stem cell differentiation. And here we have some images taken on the prime surface plates of embryonic stem cells forming spheroids. And as you can see, in less than a week, these uniform spheroids were produced using the M-shaped wells. And here, a study published in Nature Communication reported on the advantages they found in culturing with the ULA plates. They found more expression for retinal neural tissue markers. And when comparing the 3D culture grown in Matrigel to growing the progenitor cells in ULA V-bottom-shaped wells here, they also reported on the ability of human embryonic stem cells to self-organize. And what they uncovered is the formation of ciliary margin-like zones, as you can see in that picture there. And over a period of 50 days, you see the bright field view image. And this continuous layer of differentiation that is the stratified neural retinal culture. So in cancer research, 3D spheroids have helped researchers take a closer look into their drug resistance. It's pretty well known that patients exhibiting tumors and receiving chemotherapy often develop resistance to drugs, and that they were once effective and then become not effective. And according to the NIH, this is true for almost 70% of patients who exhibit solid tumors. So, and in hematological tumors as well. Using 3D spheroids, researchers have found that tight junctions proteins play a role in inhibiting the penetration of these anti-cancer drugs. As you can see, these images of the human lung adenocarcinoma cells, these spheroids are stained for their tight junctions, the claudin 1 and 3, and then ZO1. And these images were provided by the McHale lab, and they were obtained in PrimeSurface plates. And even when the images are merged, you can see that clearly the consistent overlap of this, these tight junctions and the spheroids. In other aspects of cancer research, 3D spheroids can aid in drug discovery of solid tumors. It can be done by optimizing high throughput drug screenings. In this image that we have up here, of a tumor versus a multicellular tumor spheroid, or referred to here as the MCTS. In this case, and you can see the similarities in differentiation between these two models. And they feature the same differentiation, the same microenvironment. So the idea here is that the 3D spheroids can be more predictive of the in vivo anti-tumor efficacy than in other cellular models. And by creating this more complex test system to gain deeper insights into the effects and the function of these molecules in a cellular context, we can optimize for preclinical, pre-animal selection of the most active molecules and effectors, and therefore creating a more successful preclinical and pre-animal testing results. So outside of the body, these cells are also always actually susceptible to any kind of contamination or stress and death. So in mimicking the proper mammalian environment, an incubator has to provide a stable and a uniform, warm, and a humid environment, while also mitigating contamination that these conditions attract. Optimization of cell culture conditions is very critical in ensuring that powerful experimental reproducibility, which may help to find new therapies for cancer and other diseases. So the central principle of our incubator design is that we have the best cell health in mind. And when it comes to incubator equipment, delivering the most in vivo-like environment starts with a few simple but important design points. At the top here, we have what I like to call the brains of the incubators. In order to provide the most reactive environment, or the smartest, we need an intelligent an accessible microprocessor interface that controls the sensors for the temperature, gas, and all the other diagnostics. So our CO2 sensors provide the real-time recovery and accuracy without overshoot. While the direct heat and the air jack system, along with the two independently controlled heaters, provide a consistent and uniform temperature throughout the entire chamber. And moving forward, when it comes to contamination control, which we know this environment attracts, we do have several layers in place to prevent contamination. First, our IncuSafe copper enriched stainless steel alloy provides a passive germicidal protection without any rust or corrosion. And this enables us to mitigate contamination while remaining easy to clean. And then we have our Safe Cell UV light. This features a unique sterilization system, and that safely prevents contamination at the humidification source. This light is programmed to destroy contaminants in the water pan after every door closure. And this is where all air is circulated and therefore humidified. And our integrated shelving design will minimize the parts. This creates less nooks and crannies in areas where contaminants will aggregate. It also will maximize the interior space of the incubator itself. And then we have our condensation control system. So what this does is it draws any excess moisture away from the interior chamber by way of the Peltier effect. So it's just very, very slightly cooler. And so it draws all that excess moisture away. And so this dew stick then returns the excess moisture to the humidifying pan. And again, preventing contamination or possible areas where contamination would be attracted. So beyond passively mitigating any of the contamination, an incubator must actively remove all the contaminants from the environment. This has to be done to ensure that the safe cell grow throughout. So you may have heard of hydrogen peroxide vapor as an effective decontaminant in hospitals and healthcare settings. The hydrogen peroxide vapor destroys the contaminants by oxidation. And what this does is produces a 6-log yield reduction in all contaminants. By utilizing the power of vaporized hydrogen peroxide and ultraviolet light, we've been able to provide the whole full contamination that can be achieved in less than 3 hours. There's no need to remove any of the components or the sensors, so it's total convenience. And this is just creating a more efficient way to follow regimented decontamination protocols. And here, I just wanted to give you a look at our Cell IQ line of incubators. These feature those different layers of control and security. And that concludes my presentation here. I'd like to thank everyone for their time.
How does the UV light decontaminate without harming cell culture?
Okay, I always get questions about the UV light. So there also is a handout that shows an illustration in the handouts, and it is called the HEPA versus UV feature. So you'll see there's a picture of the UV working within the incubator itself, and there's a plenum behind the incubator that shields all of the cell culture from any possible UV radiation and anywhere else in the environment. What it does is it just focuses in all of those UV rays, those sterilizing UV rays, down to the watering pan, and that's where it acts and disinfects. And then all the air that's circulated and humidified in turn goes through this process. So it's able to go ahead and use this technology without harming any of the cells and just create that clean airflow in a titration. And again, you can refer to the handout if you have any other questions about that too, and it has a nice little illustration there.
Great, thanks very much, Holly. Our next question is, for what applications are slit well plates used?
Oh, okay. So the slit well plates here, those are usually used with IPCs or things that for regenerative medicine, things that need a long-term growth. Like for instance, like the IPCs, those cells grow and differentiate like after a few days to weeks. So for that long-term growth, it just makes that step really easy, and that's what was intended with that particular plate.
Great, thanks very much. We have another question that says, how do I know which shape well to use?
Great question. So they have found that depending on the cell's tendency to aggregate, a different well shape is preferred. And there's a growing list of publications that they have found where their cell lines prefer different shapes depending on their aggregation as well. Like in this PowerPoint that I went over in the presentation, I was discussing that a lot of the stem cells were preferred the M-shaped or in some cases the V-shaped, but then the cell cancer breast line, the breast cell cancer line, had trouble aggregating, so they really did the best for tight, nice uniform spheres in that V shape. So it really would have to be, to answer your question fully, it's a little trial and error and it's a little bit of reading. And also you can always come and contact us and we can reach out and give you some examples in different publications referring to which bottom wells were preferred in different cell lines.
Great, thanks very much. We have time for one more question, which is, how often do you replace the UV light?
So the UV light, and I do think this is also in that handout with the UV light versus the HEPA. So it's 5,000 hours of use. So it's about 7.5 years, depending on. And the UV light time automatically extends according to the increase in cumulative lighting time. So that's important. It's always remaining effective. And there is an indicator of life that you can look and refer to for replacing it. But again, the average would be about about 7.5 years total.
This webinar video has been made possible by the PHC Corporation of North America. For more information, visit our website at www.phchd.com/US/biomedical. And for specific information on PrimeSurface 3D cell culture plates, visit www.phchd.com/US/biomedical/PrimeSurface.